hopefully by the end of this presentation, you'll have some answers to some questions. So what are emerging organic contaminants? And what on earth does the acronym PPCP stand for, which is pharmaceuticals and personal care products? And have an understanding of how they get into the environment, how we analyse for them, when we find them, what kind of concentrations we find, what kind of impacts might these chemicals be having in aquatic environments, and how we might go about reducing the impacts in aquatic environments. Can everyone hear me okay? Okay, that's great. So what are emerging organic contaminants? Now these are compounds that are not routinely monitored for in the environment. However, they are of interest. Either these are newly developed compounds, so for example, this might be a new pesticide, or it may be a new preservative in a product. They are often compounds which have only been recently detected in the environment due to improvements in our equipment. So the analytical equipment gets ever more expensive and ever more fancy, and that means that we are actually now able to find some of these contaminants in the environment. They may have been there for a long time, as you'll see as we go through, but it's only now we have the technology to be able to find them. Or it may be a compound or a chemical that because of new information, new toxicological information, we now have a reason to be concerned about it, and it's now of some interest. So the ones I'm going to focus on tonight are pharmaceuticals and personal care products, or the PPCPs. Should also point out that household cleaning products are now on the watch list for being sources of emerging contaminants, and some food ingredients, including artificial sweeteners. So personal care products. These are everyday chemicals. These are things that we all use every day. So this is our hygiene and personal care, cleaning your teeth, having a shower, those sorts of things. So disinfectants, fragrance ingredients, musk fragrances in particular, insect repellents like DEET, preservatives, the parabens are an example, UV filters that are added to sunscreens to protect us from the sun's rays. Now these UV filters are often added to a lot of makeup and a lot of other sort of skin creams and products to improve their shelf life for stability. And of course there's ever increasing numbers of antibacterial compounds, so things like triclosan, tricloban, products like Dettol or wipes that are antibacterial this and antibacterial that. Now the most of these are used externally. So when we have a shower or a bath or whatever, they wash off and they enter the sewage system unchanged. For pharmaceuticals, I'm going to stick with the World Health Organization definition. So we're talking about the kind of products that your doctor would prescribe to mitigate or treat some form of disease or some symptom. It also includes things like aspirin or paracetamol or things that we might buy over the counter. I should say that around 150, maybe getting closer to 200 now, pharmaceutical compounds have been monitored in the environment. And about 18 to 26 new therapeutic drugs are approved annually by the US Food and Drug Administration. So why are we now worried about them? Well, the reason we're worried about them is because these things are biologically active by design. They tend to be water soluble. In many cases, they're now considered to be ubiquitous contaminants, which means that everywhere we go looking for them, particularly if sewage has been discharged, we will find them. They are only partially treated or removed by wastewater treatment processes. They can also tend to, as this compound here, which is avoparsin, this is an example of an antibiotic, they can be quite large, complex molecules. And because of the number of different groups on here, this is a bit of a shapeshifter. So depending on what's going on in the environment, it can change its format slightly, which makes it even harder to remove. What's been of a big issue overseas is that some of these compounds have now been detected in drinking water. So now people are asking questions about, well, we know it's a lot less than the therapeutic dose, but is this still okay for people to be drinking? Is it still okay for people who don't have high blood pressure to get a low dose of a blood pressure medication? And for many of these, we're also concerned because they can have endocrine disrupting effects, which mean that they can work on your body's hormonal signaling system. Now, most of the work that's been done until now overseas has focused on freshwater environments, and that's because these compounds were initially found in smaller rivers and streams in the US and in Europe, where there were sewage discharges. So most people have sort of said, well, we know they're in wastewater, and we know that they're in freshwater, And when they get into the marine environment, they're possibly not going to be such a big deal because you've got such a big dilution factor. That's turning out not to be the case. Also, when you think about it for New Zealand, our population is predominantly coastal. Most people live within 10 kilometres of the coastline, and 90% of the sewage is discharged into the marine environment, with a small amount going into estuaries, freshwater, or onto land. And of course, our coastline, we all consider it a treasure, and it is a big source of income for New Zealand, both through tourism and through aquaculture, fisheries, those sorts of things. And of course, these coastal ecosystems are under increasing pressure from urbanisation, expansion of aquaculture, climate change, and a range of sources. So this is one of the reasons that I'm focusing my attention on our coastline rather than in fresh water, because I think for New Zealand, for the kind of contaminants I'm going to talk about, the coast might be more important. So these emerging organic contaminants, where can they come from? Can be a big problem if they're used in livestock treatments, and that's particularly been a problem overseas where they have used human pharmaceuticals in feedlots and with animals. Don't forget, many of the drugs we use on people also might be used on companion animals. We take them ourselves. There has been some evidence in India and as well as in the US of high levels of some of these pharmaceuticals from manufacturing processes. That was quite interesting because the impression was that this would only be a developing country problem, and in some cases downstream of wastewater treatment plants in India, you can get a therapeutic dose of antibiotics by drinking the water. So they're losing product really. However, they have recently found in the US that some of their pharmaceutical manufacturing plants also put out a large amount. However, for the most case, it's what we take. They end up in our wastewater. We can get leakage. Now, that's probably a bigger problem in Christchurch at the moment than we would like. And then they can end up in receiving water. And in some countries where they take the solids that are produced from wastewater at the end of wastewater treatment and they use them as a fertiliser on land, then you can end up with the pharmaceuticals in land. They can then leach onwards. But this isn't a new problem. The first report was in the 1960s where they said that water had estrogen-like properties from wastewater. And the first report of a pharmaceutical in the environment was in 1976, which was clofibric acid. And in the 1990s, Swiss and German researchers also detected personal care products and pharmaceuticals in wastewater and surface water. And then it was all very quiet for a while. And one of the reasons it was quiet was because our analytical equipment wasn't good enough. We couldn't really detect the kind of environmental levels that we would need. And so there's been recent advances that mean that we can now measure these in environmental samples. We're still going to need some more advances to be able to detect some compounds, and particularly if we're wanting to detect these compounds as they move into organisms and biological matrices. So what happens when these compounds, so preservatives, additives, ingredients from our toothpaste, our shampoos, sunscreens, end up in the environment? So the first thing that can happen is that they can be degraded in some way. So that might be by microorganisms. It might also be by being uptaken by an organism. Sunlight can do a lot of the degradation for some of these compounds. However, many of our pharmaceuticals and personal care products have been designed to be stable. They're designed to have a long shelf life. They're designed so that when people take them or when they're used, they are what they are meant to be. They're not meant to undergo degradation processes. Some of them, depending on their properties, will stick onto sediments, and some of them will be taken up by organisms. Many of these things will have relatively short half-lives in the environment, but that doesn't really work if you're a fish that lives downstream of a wastewater treatment plant that is constantly, because of the population that lives there, constantly has about the same concentration of a contaminant that you have a problem with. Then it doesn't matter that it doesn't last very long in the environment. If it's constantly flushing through your home, then you've got a problem. Working with these compounds is exceptionally challenging. These are complex extraction and analysis techniques. It can take us up to 4 days from when we collect a sample until when we have the sample ready to be analysed. One of the problems with this is we now have a very large range of compounds. So this can include the parent compound, anything that's been metabolized. This is particularly important for things like pharmaceuticals. When we take them, they may change in our system, and the way we excrete them is not in the same form that they came in. And there may be, if they undergo some form of degradation, whether it's by microorganisms or sunlight or some other process in the environment, that we've now got actually different chemicals to be looking for. Choosing which ones can be quite tricky, and there are now some labs in the US who may be looking for 300 or so of these compounds in one go. One of the other problems is I talked a little bit about shape-shifting. Depending on the pH, so how acidic the solution that they're in or not, they will change. So they may be neutral species, so they don't have a charge on them, or they may become a positive species, or they may become a negative species. Now this gets tricky because our techniques are either designed to deal with positive species, negative species, or neutral species. Another problem is many of these compounds are water soluble. So unlike some of the pesticides of the past, things like DDT, which is actually relatively easy to analyse for compared to these, because it's not very water soluble, it'll stick to almost anything. So it's really easy to get it out of water. But these things like to stay in water. This one here is a big problem. We need a standard. So a standard is we need a pure compound so we can compare back to. We use that pure compound to make up our analytical standards, which is how we determine concentration. And often, some of these metabolites and transformation products, which may actually be more toxic than the compound that we started with, we don't have these reference materials. We can't get them. We either have to make them or say, yes, we know that we're not accounting for everything, but that we can live with that. The other issue is these are at ultra trace concentrations. These are at really, really low concentrations. And this costs a small fortune. I don't charge this much, but I do know that one of the commercial laboratories, when asked how much it would cost for one sample, it was $1,600 a water sample. So you can see the costs can add up very, very quickly. So just an idea, give you an overview of a sample, of a process. We will collect our sample. We always collect it in a clean, dark bottle. Now, clean to me probably doesn't mean the same as what it does to you. Or actually, I just say how I'd like it cleaned, and the poor PhD students usually end up having to do it. Clean means washed with detergent overnight. Rinsed 10 times with tap water. Rinsed another 10 times with pure water. And then rinsed several times, maybe up to another 10 times, with expensive solvents. So we've made sure that there is absolutely nothing in there that could contaminate what we're doing. Then it's clean. The reason for having a dark bottle, like these ones here, is we don't want any sunlight to degrade. So when we collect the sample, we want it to be as we collected it. We preserve it by adding some sulfuric acid, and then we store it at 4 degrees. And the reason for the 4 degrees is because, well, often we're dealing with sewage, which you could imagine has got a lot of bacteria in it. We don't want to create a health risk for the rest of the laboratory, and we also don't want our sample to degrade while we're working with it. We then use what's called solid phase extraction, or SPE. So this is a manifold here. We have these cartridges here. These are the expensive part. These are Teflon lines. So we basically suck the sample through under vacuum from here onto there. So hopefully our compounds, and this is why it gets tricky about knowing what form they're in, will stay on the cartridge, and we can throw the water out. Then we can come along using solvents, so things like dichloromethane and hexane, acetone, other ones depending on what we're doing. We can then extract them off into a small vial. Now this is great because we can start off with 10 litres here. I think that's the biggest we've done. And by the time we're finished, before we get to analysis, we're down to 0.3 of a mil. So we can get concentration factors of several thousand times, which is why we can work at those ultra trace levels. Now, many of the pharmaceuticals and personal care products, people are now using liquid chromatography, mass spectrometry, and I'll talk you through that. I think it's a really neat technique, and I want one, but it costs $750,000 to get what I would like. So I think it might be on my wish list for a while. Well, true, maybe that's how I could get one. Then what we do is we analyse our sample. At the same time as we analyse our sample, we take solutions containing pure compound, where we know exactly how much was in there, to form a calibration. So we then have a response for our standards, so we know what the response is, and then we can compare the response from our unknown samples to that, and we can calculate how much is in there. Should also say one of the things that adds to the expense of this, because we're working at such low levels, the level of quality assurance and quality control samples that you have to prove that you knew what you were doing means that most of the time, about 50% of what the students are doing is just quality assurance, quality control, and not actual samples. So proving we know what we're doing. So, LCMS. So what we do is we use liquid chromatography to separate. So basically we have like a tube or a column. So I'm just going to flip through a couple. So we've got an instrument that looks like this. This is an LCMS. In here is the column, which I'm just talking about. We pump a mobile phase, a liquid phase, through the column. Now this column has what's called a stationary phase, or it's got sort of like little beads in there. And on those beads is another surface, which again, we want our compounds of interest to stick to. Hopefully, they will all have different affinities for that stationary phase, so they separate out. A bit like people going down a buffet line. You know, some people will stop with the salad and some people will stop with the dessert. So you kind of spread it out along the buffet line, if that makes sense, because they've got different likings for the stationary phase. The ones that really, really like the stationary phase, like this case here, which is gemfibrozil, comes off last. This one here, metformin, which may be familiar to some people in the audience, doesn't like the stationary phase very much, so it comes off first. This along here at the bottom is time, and hopefully we've got these nice separated peaks. Each one of those peaks in this example is a different pharmaceutical. And up the side, we have a response from our detector, which gives us some idea of how much is there. What the really neat bit at the other end is the mass spectrometer. So basically what this is, it's a detector, so it actually sees our compounds, for want of a better word. We take each compound as it comes off, one in a row, break it up, and form these fingerprints. What we can do is we use what's called the mass to charge ratio. So hopefully everything's singly charged. We look, and we have a fingerprint like this. So this is for caffeine. Some of us will have had that drug today. We can pick one of these ions. So this is the fingerprint of all of caffeine. We can ask the instrument just to look for one of these ions at, coming back here, a specified time. That's how we can get down to the really, really low levels. Why I'm so excited about this is this has been the huge leap forward for being able to do these pharmaceuticals. The fact that they've actually been able to, the mass spectrometer works inside a vacuum to be able to do what it does, and the fact that they've actually been able to link a HPLC column with liquid into a mass spec and into a vacuum is pretty cool. Well, I think it is. Some of the students don't quite understand my excitement with it. Okay. So before we start talking about the kinds of concentrations we might see, just a little bit about units. Analytical chemists tend to talk in parts per million, parts per billion, and parts per trillion. Parts per million, that's easy. So that's a milligram per litre, and that's equivalent to 1 minute in 2 years. Parts per billion, not quite so easy, micrograms per litre, and it's about 1 second in 32 years. Many of the compounds or the chemicals I'm talking about tonight are in the parts per trillion level, which is nanograms per litre, or 1 second in 320 centuries, and I have checked this, it does work, or 1 drop in 20 Olympic swimming pools. The techniques I've just shown you can find these drops in the 20 Olympic swimming pools. That's the exciting part about it. Do we need to get this low? Yes, we do, because many of the contaminants are at this level and they are having effects at this level. So, just to give you a little bit of data, this is from international data. This is for sewage treatment plants. So what we have here is compounds, so some of these I'll go through them, what kind of class it is, and then the kind of concentration ranges that they're finding. So these are micrograms per litre, these are parts per billion. Where it says less than LOD, that means limit of detection. So what we have to do when we use these instruments is set a limit that's kind of our baseline. Below that we say we can't see it, and above that we say we can see it and we can confirm its presence. So we've got some lipid regulators there, 3 anti-inflammatories, a beta blocker, carbamazepine, and triclosan, which is an antimicrobial. So this is the one that when you get an antibacterial soap. So as you'll see, here we're talking parts per billion range. As we move over to our surface waters, now these will be mainly fresh water, we're getting into the nanogram per litre range. So here again we have triclosan, a disinfectant. Methyl triclosan is a degradation product of triclosan. It's bacteria make this one. Tricloban is another disinfectant. These ones here, musk ketones and musk xylenes, the other things that often make soap smell nice. DEET, an insect repellent. Parabens, which are preservatives. And these ones here, the UV filters, they are the organic compounds in your sunscreens that absorb the UV rays instead of your skin. So why are these things getting in the environment? We've got sewage systems, so why isn't that dealing with them? Well, the reason being is that sewage treatment plants are designed really to deal with 4 things. First of all, they want to get out the big chunky bits and the false teeth or whatever the toddler put down the toilet. Then we want to get rid of suspended solids. We want to get rid of biodegradable organic material because that's what causes problems in waterways. And we want to, of course, make sure people don't get sick, so we want to reduce pathogens. This is what our wastewater treatment plants have been optimised to do. Now, for many of the other contaminants that have got into sewage, metals, some of the persistent organic pollutants, the organochlorine pesticides, this still worked reasonably well because things stuck onto the solids. So if we took the solids out, then we removed the contaminants so we had no problem. But now we're talking about things that are water soluble. So they tend to stay in the water. And many of them, as I said, have been designed to be reasonably stable. They're not going to undergo degradation because the pharmaceutical companies want to make sure that when you take a drug, you are taking what they think you're taking. So this picture here is just an example of a wastewater treatment plant. This one's actually in Tenerife. It's got one of the best views I think I've ever seen from a wastewater treatment plant. And this here is in their lab, and this is actually showing you what most wastewater treatment plants do. So this is influent, this is effluent. So basically you want to go from sewage with stuff in it to a clear liquid at the end. That's how our wastewater treatment plants have been designed to work. But of course, if you've got something that stays in the liquid, then it may still be here when you get to the other end. There are different levels of wastewater treatment, and depending on where you are in New Zealand, there will be a varying degree of wastewater treatment. So we've got preliminary, which is basically taking out grit and plastic ducts or whatever went down the toilet. Then we've got primary, which is sedimentation and basically just settling out the solids. Secondary, which can be a biological process, like here at Bromley, so that's where you might have activated sludge or some form of bioreactors. Or you can also, in the more urban areas, may start having tertiary treatment. Now these ones were usually brought on to remove nutrients, so removing nitrogen or phosphorus. However, we're now starting to have to look at what is beyond tertiary, and if we're going to have high-level treatment plants, what would they need to be able to do to remove some of these contaminants of concern? Now, until recently, I think Wellington really only got somewhere around here. So before they put in Moa Point, they just macerated it and put it out in the sea. And I think that was also the case in Dunedin. So this is kind of the level where many of New Zealand's wastewater treatment plants stop. And of course there will be other areas along the coastline where people have batches and septic tanks and things where the level of wastewater treatment could only be described as variable, and I'm not sure that treatment is often the right word. Sometimes it's just there. And it's not uncommon for people to put septic tanks, put a hole in the bottom of the septic tank so it works better. Better meaning it went away rather than it actually treated anything. Okay. So when we start thinking about our pharmaceuticals and personal care products, what are the adverse effects? And the honest answer is for many things it's largely unknown. Many of the pharmaceuticals were only tested in terms of what they did to people. We may know something about what they do to animals because they may also have been used on animals. And I'll show you some examples a bit further on where things have gone a bit haywire. The good news is that we haven't had that many acute effects. So acute effects means usually exposed to a very high concentration of something over a very short time. So kind of dead, maybe, or very, very sick very quickly. The effects are more likely to be chronic because we've got this sort of long-term, low-level exposures. So some organisms may be exposed over their lifetime. And I've mentioned this before but didn't define it. Pseudopersistence is a big problem. As I said, many of these compounds may not necessarily last in the environment for more than a couple of days, some may be hours. But for organisms that live downstream of wastewater treatment plants, where you've got a population generally all taking about the same kind of dose if you average across the population, and it's constantly being put out, then you're constantly being exposed to about the same concentration. So it doesn't really matter if it lasts for a long time or not. This one here is the one that everyone is struggling to get their head round. What do we do about mixtures? Most of the studies that have been done to date have been done on a single compound or a single contaminant. So we don't really know what happens with a cocktail. We don't really know how to assess that. We don't know which of the chemicals are the most important to put in the cocktail if we do a lab test. And we don't know how to work out yet whether some things may cancel each other out. Some things, if you've got two of them, you might get a worse effect, and some things you might just get the same effect of, two things, 1 + 1 you get 2. And there really has been limited research undertaken to date. And one of the problems is many of the toxicologists, they're just overwhelmed. I say we, I do a little bit of tox. I'm more on the analytical end. We have very large numbers of compounds that have been detected in the environment. Starting to think about how to prioritise these. What is the most important thing to be looking for? What are the most important adverse effects to be looking for? We're changing a lot from, as you'll see, from the old style toxicology, which was crudely called kill 'em and count 'em. You basically did an effect concentration 50, you worked out when 50% of the organisms were dead, and you said, all right, we'll just lower that a little bit. We've moved on from that, and as you'll see, some of the effects we're starting to see are quite subtle and are often novel. So what have they seen? There has been some toxicity reported, particularly in fish. Fish, I think, tend to be quite affected by quite low levels of some of these compounds, and levels that people initially wouldn't have thought would be a problem. The big one from a human health point of view, and I'll talk more about this in a couple of slides, is the development of antibiotic resistance in pathogenic bacteria. So this means that some bacteria have developed the ability to deal with antibiotics. And this is a problem if it's pathogens, because then we can't treat disease. A variety of different organ damage, kidney damage, liver damage, damage to the gills has been reported in fish. There's been some genotoxicity, so you start seeing changes to the genetic material of the organism. This is the one that really interests me, is behavioural effects, and I'll go through that in a little bit more detail. Endocrine disruption in fish is quite important, with changing their hormonal signalling, and quite a few studies are reporting effects on algae, and of course algae are the bottom of the food chain, so if you wipe them out in an aquatic ecosystem, you may have a bit of a problem. How do we measure these effects? Some are done, and some of the early work was actually found by field work, so people looking at organisms downstream of wastewater treatment plants. A lot is now done in laboratories. These little guys here are zebrafish, and they are very commonly used. They have share some genetic material with us as well. So they're often used for these kinds of studies. Now, the traditional endpoints were, can I kill it? Does it grow or not? Does it reproduce? But as I said, those are not necessarily the right endpoints to be looking for anymore, as we struggle to work out what to do about some of these contaminants. We might use biomarkers. So these are changes in blood, changes in chemicals in blood or other tissues, where we can say, actually no, we've seen an effect. And sometimes we might see changes to biomarkers much earlier. So very similar, if anyone's had a blood test from the doctor and they do a whole lot of biochemistry screens on you, they may be looking at your liver function or your kidney function, same kind of process. There's a whole range of what we call the omic responses. So does it change the kind of proteins this organism creates? Does it change its genetic material? Some of the studies will be done in vitro and some will be done in vivo. The difference there is looking at whether it's done with cell lines or whether you do it in a whole organism. So sometimes we may just use cell cultures if we're targeting specific things. Histopathology is still part of the kill 'em toxicology. So in that case, you may expose organisms to a range of your contaminants, and then they necropsy, take them apart when they're dead, and have a look at the different organs and see whether you can see any effects. And of course, we will also be looking for what happens with a very short, sharp exposure to a high concentration and what happens to over a longer term. And for some of them, like these poor little guys here, they will do intergenerational studies. So these guys have a reasonably short short lifespan, so they can do multiple generations and actually see if these effects persist. So invertebrates, Daphnia magnia, little water fleas, plants and algae, quite a lot done with microorganisms. And then the poor zebrafish and rainbow trout have probably been studied to death in many places, literally. However, one of the other things people are starting to look at is what do we do for marine environments, what do we do for estuarine environments. So people are trying to develop other model organisms that they can use. As I said with algae, this is a real problem. Algae are sort of an early warning system that you may be having an effect in an environment. If you start changing either who lives there or how much of algae live there, then you know you might have a problem. So we're talking about species diversity, we're talking about the range of species that live there. If you have too much of some personal care products, in particular and some pharmaceuticals going in, they can change who lives there. They can change the nutrient cycling, which is quite a problem. They can also just stop the algae from growing altogether, which means everybody above that who eats that will be hungry. Now this picture here is actually of a toxic algae. This is a cyanobacteria. And the reason I've put this in here, this picture was taken by Francine, one of my PhD students who worked on toxic cyanobacteria, is there's been a very recent report come out in the last couple of weeks that they've, through a combination of field work and work with artificial streams in the US, they've found that downstream of wastewater treatment plants where there's increase in triclosan in the sediment, which is one of the antimicrobials in your toothpaste. It's also in some of those wipes and things, and many of the hand soaps. That you can actually, these guys, some of the toxic cyanobacteria, are able to cope with the triclosan a little bit more, so you get more of them and maybe less of some of the other species. Now, of course, because these can produce quite nasty toxins, that's not really something we want to see happening in our waterways. This here is another example where they looked at 3 different personal care products or pharmaceuticals. So we've got ciprofloxacin, which is a fluoroquinolone antibiotic, triclosan, and tergitol, which is a surfactant, and natural algal community. So what we've got along the bottom is the different concentrations of the personal care products. Up the side, what they've done is a mixture of what they call genus richness. So they've counted the number of different types of algal species that are there. As you can see, as you increase the concentration of both of the compounds, you're actually starting to see a decrease in your number of algae that are there. Now, of course, this is particularly problematic, as if you're going from here to here, you start getting more of the toxic ones or more of the ones that aren't so beneficial. So that's one of the examples of effects on algae. Now, diclofenac, Voltaren, commonly available over the counter. Some people will be prescribed it, some people will rub it on their skin. There's always showing people rubbing it on their elbows. This study here is quite important because what they did was they exposed, again, the poor rainbow fish to a range of different concentrations of diclofenac. And then they looked at cytological or changes in cells in the liver, kidneys, gills, and intestines of the fish. The lowest observable effect concentration, or LOEC, was 1 microgram per litre. Now, why this is important is this is an environmentally relevant concentration. This is the kind of concentration that we would find in the environment. Now, some of you may be aware that diclofenac is being considered at the moment as to whether they will ban it for human use because of effects on kidneys. So that's one that's, that sort of feedback between human effects seen in humans and effects seen in organisms. This is the one that really fascinates me because it's one that I've only become more recently aware of, is changes to behaviour. So there's a whole branch of ecotoxicology, which I hadn't heard about before, called behavioural ecotoxicology. So what this is looking at the effect on how an organism behaves, how it feeds, maybe how territorial it is, maybe how long it lives in an environment. So these here are Japanese Medaka fish. I always look the fish up now because I'm always curious, and they're usually something quite small. What they did was they again fed them triclosan, diclofenac, and carbamazepine, Carbamazepine is an anti-epileptic drug. Now this one's quite important because it's not removed by wastewater treatment plants. It sails straight on through and is quite persistent in the environment. What they found is it actually changed the feeding performance, how often they fed and how long they fed for. Some of you will have noticed that in the brief for the talk, I talked about finding antidepressants in fish brains and does it matter. And that was found. There was a study done in the US where they looked downstream of wastewater treatment plants. They did a big US-wide study of what was in fish and what was in the water and what was in the sediments. And they did find Prozac in some fish brains. And then that left a lot of people thinking, well, does that matter? What does that mean? Well, they have found that if you have environmentally relevant concentrations of some antidepressants, so by environmentally relevant, we mean the kind of concentration that you actually find in the environment. You get, the juvenile fish are not so good at predator avoidance. They're just not scared of predators anymore. Which sounds kind of funny, but for your fish population, I mean, I guess this is a bit like teenage boys maybe, if they're not scared of being eaten, then that's not so great for your population either. And they've also found that compounds that can act as estrogen mimics can alter aggression and territorial behaviours in fish. So some of these fish, they don't protect the areas. Some other examples that I've heard of are some fish species, the male fish looks after a nest. They just don't do that anymore. Some of them, they just don't do things that they normally would do, which can eventually have a very severe effect on your population. Some people have also been looking at behavioural changes in birds and in other larger organisms. Of course, the big one that's sort of pushed everyone's buttons, because this is probably how this whole problem of pharmaceuticals and personal care products was first found, was endocrine disruption. So our endocrine system is a complex mixture of sort of hormonal signalling that tells our body what to do and when to do things. So we can get endocrine disrupting effects that are either estrogenic or anti-estrogenic, so we can actually make the system believe it has more estrogen or less estrogen. We can also muck around with the male hormones. So things can either be androgenic or anti-androgenic. So more testosterone, less testosterone. The one that they're getting increasingly concerned about is effects on the thyroid system. And why that's important is because this has a lot to do with how organisms regulate temperature, how their immune system functions. So this one is particularly problematic. So what can happen is, remember we're talking about some of these contaminants that are water soluble, they're designed to get into cells. When they get into cells, then they can either link up with the hormone receptor and the body goes, great, all my machinery's in place and something will happen. Or it can get into the hormone receptor and make it not work. So the body says, oh, nothing happening, so I don't need to do anything. So they can actually really muck around with the signalling in a body. So what kind of endocrine disrupting effects have been observed in wildlife? Now this is going to depend on the timing. Many of these are considered to be particularly problematic if it's at the stage where you're developing. So the developing fetus is considered to be the most sensitive. So this might be at the egg stage, larval stage, something like that. So the original ones that they found were to do with intersex and altered sexual behaviour. So intersex is when, and this is particularly a problem with fish, another new term I learned from the toxicologists, can be termed what's called sexually plastic. So they're very easily converted between male and female, and for some fish species that's part of their life cycle. So what happens here is you've either got a male fish starting to take on female characteristics or a female fish starting to take on male characteristics. Again, this can do interesting things for reproduction. If for some contaminants being exposed during development may change their reproductive organs. In some cases they've seen skewed sex ratios, so you might get more males or more females. These ones here around the modified immune system and altered thyroid. Some of these are starting to be more important and may link with some of the behavioural stuff, is starting to see brain and neurological problems. So there's a wide range of effects that have been observed in wildlife. Many of these have been observed in fish. Okay, so starting to talk a little bit more about our intersex, so thinking about, are these fish, are they male, are they female, or are they both? So the first way that they do it is by, and I haven't got the pictures because they're a little bit gory, is by histopathology. So if you look at a fish and you look at a male fish, it may have eggs in the testes. You can also have it go the other way, where a female fish is starting to take on characteristics of male fish as well. And so you're starting to see changes in what their gonads look like and how they develop. The other way you can do it is, particularly for estrogenic compounds, is you can take blood from your male fish, and vitellogenin, which I always find a mouthful, is an egg yolk protein precursor that's produced in the liver of female fish. This needs to be produced to make eggs. So if you have it in a male fish, then you know that possibly something's not going quite as well as it should. A lot of the issues around pharmaceuticals in the environment came up to do with 17-alpha ethinyl estradiol, or EE2. This is the active ingredient in the contraceptive pill. Just to give you an idea of the kind of concentrations, this is in the US, Germany and Netherlands and the UK, we're talking at 0.1 to about 15 nanograms per litre. So we're talking handfuls of drops at the most in our 20 swimming pools. The concentrations that have been reported to cause adverse effects in fish, this is alteration of sex ratios in zebrafish, 1 nanogram per litre. Reduction in hatching success, 0.2 nanograms per litre. This is inducing that egg yolk protein in minnows, 1 nanogram per litre. These numbers here fit in here. And also they have seen behavioural changes from exposed to ethinyl estradiol in zebrafish, where that altered their aggression and shifted individual social status and reduced reproductive success in male zebrafish. So the male zebrafish need to be quite aggressive for the girls to think they're nice, and if they're not as aggressive, then they have reduced reproductive success. This here is a very famous study that was undertaken by Karen Kidd in Canada. So up in the northwest of Canada, they have what they call the experimental lakes area. Unfortunately, this has just been closed due to government cutbacks. So they've got a series of lakes where you're allowed to do experiments. So what Karen's group did was they added in low doses of basically the contraceptive pill of 17-alpha ethinyl estradiol into the water. This project was meant to run for several years, but they actually had to stop because they got too near extinction of the species. So they saw intersex in males and altered egg production in the females. And basically they collapsed the population and they kind of had to go, okay, we stop. Now, several years on, these are actually starting to recover, but this is probably one of the most famous kind of big widespread studies that's been done on the effects of pharmaceuticals. I should also add that at the conference that I was at a couple of weeks ago, I learned of a new and unusual use for 17-alpha ethinyl estradiol. Up until now, we'd assumed that everything that went into the wastewater treatment plant would have been taken by a woman and would have been excreted. However, apparently, if you mix the contraceptive pill with your shampoo, it's also a cure for baldness, which has left a whole lot of people wondering and shaking their heads as they're starting to do the risk assessments and calculate how much you should be expecting to find, that if this craze takes off, we may have a lot more and in different forms than we thought of. Okay. One of the big concerns as these contaminants get into the environment, and if they are persistent enough in water, is do they get into organisms? And do they get into organisms at concentrations that are high enough to be of concern? So these guys down here, this is from Rathi, another one of my PhD students who actually worked on cadmium, but these are some of the greenlet mussels out in the field. These guys are filter feeders, so they take up very large concentrations of water. So as they take up very large concentrations of water, if there's any contaminant in there that they can absorb into their tissue, then they take it up. So they're a bit like my SPE cartridges I showed you before, except these guys do thousands of litres of water. So we start getting concerned if we can start finding things in shellfish. Now, we use a term called bioaccumulation. So that is an increase in the concentration of a compound in an organism relative to where it's living. So if we can find a higher concentration in the organism than in the environment, we know it's accumulating it. To accumulate it, you need to take up your compound faster or your chemical faster than you excrete it or metabolize it. The extent of our bioaccumulation will depend on the contaminant structure. So depending on the building blocks of our different chemicals, some of them will be more persistent in the environment and some of them might be more likely to be taken up by organisms. This one here is one of the first where I've seen quite a few studies of bioaccumulation of UV filters in fish. I've seen quite a few studies of bioaccumulation of UV filters into mussels around the world. However, this is a recent one that's come from Brazil where they found octocrylene, this compound here, in these cute dolphins here. So these are Franciscana dolphins. They're about the same size as a Hector's dolphin, maybe a little bit bigger from what I can work out. They took liver tissue. Many of these compounds will go to the liver to be metabolized or excreted from the body. And they did find octocrylene in 21 of the samples. Now these units here are LW as lipid weight. So in this case, they have worked out the amount relative to the amount of fat in the organism. What they found, which is of concern, was that these were much higher than previously measured in fish. And this suggests another possibility that hasn't really been thought about until now is biomagnification in food chains. What that means is, and some of you will be familiar with some of the organochlorine pesticides and things like mercury, that things that contaminants that bioaccumulate in food chains, you get higher and higher concentrations as you go up a food chain. So you get the little fish that is eaten by the bigger fish, eaten by the dolphin. So we can start seeing these higher concentrations. And this is probably one of the first examples I've seen of this, and this is quite of concern. So they did find some temporal differences with these UV filters and sort of linked it with times of higher usage. One of the things I'm interested in is we've got quite a lot of marine reserves in New Zealand. They're kind of loved a lot by the community. And you can have hundreds, if not thousands, of people swimming in a marine reserve all covered in UV filters, which we know can impact on aquatic organisms. This is the big scary one for me, antibiotic resistance. This is the one where there are human health implications. So what happens is bacteria can develop the ability to deal with antibiotics. So they can work out how to just make the compound not work anymore. They can look at modifying susceptible sites in their cells so it doesn't work anymore, or they can figure out how to spit it out and just get rid of it. What's the more scary about that is microorganisms are really good at now sharing this information with their friends. So once they've figured out how to do this, they can then pass those bits of their genetic material to another microorganism, which now means that that microorganism gains the ability to do that. Where this gets scary is because often this information will be in a non-pathogenic bacteria, so a bacteria in an animal's gut or one in the environment, that we wouldn't normally be worried about. But if it comes in contact with a pathogenic bacteria, then it can hand over this information, here, have this, you can deal with anything you like, which now means we've now got pathogenic bacteria that we can't treat. One of the other problems is our love of antimicrobial this and antibacterial that in a lot of our household products. There is some evidence to suggest that as we have more bacteria being exposed to low levels of these antimicrobial or antibacterial compounds, they can also develop resistance to antibiotics, which they can then share with their friends, and particularly their more nasty friends. Scarier still, other contaminants, including metals, have also been shown to trigger antibiotic resistance in bacteria. So this is considered a very serious issue by the World Health Organization, and this, when I go to the meetings on pharmaceuticals in the environment, this is the bit that scares the scientists. We're going to need to work out how to deal with this and how to prevent it if some of our everyday surgical procedures and things that we all take for granted are to continue. Okay. This is an example of unexpected effects. So we're still working out, as I've said, what are the important endpoints, what do we need to know about, what are the effects we need to know. Over a space of about 10 years in Asia, 3 of the Asian vulture species went through a complete catastrophic collapse. They disappeared. So this is Nepal, India, and Pakistan. I should say, and they were gone. And people looked at all sorts of, was it disease, was it pesticides, what was causing this? What was found out to be was actually a really nasty form of kidney failure and gout I didn't put one of the pictures in because it's too grim. These birds died a really painful death. What happened was, in India, when an animal dies, it gets left out and taken away by the vultures. And in actual, what happened was many of these cattle were old and they'd been fed diclofenac. So pharmaceuticals are quite readily available in some developing countries like India. These old cattle were being fed diclofenac or Voltaren to keep them working a bit longer. And when they died, they'd just be dumped and the vultures ate them. What was happening was the vultures that they eventually figured out, when they died, they'd been feeding on these fresh carcasses that had Voltaren in them. And so then the vultures died. Well, does that really matter? Well, yes, they'd had a big decline in species for vultures. This was problematic for some communities. Some of you may be familiar with the Parsis and sky burials, where you leave people out as well. That could no longer happen, which was of cultural significance. However, there were also some significant public health risks associated with the loss of the vultures. Because you had a culture where you leave dead animals out and they get taken away, and that was still happening, they got large increases in stray dogs and leopards and rats. Now, there's been some modelling work done that suggests that an extra 50,000 rabies infections occurred, and possibly deaths, due to the increase in dog bites from feral dogs, because the feral dog population was able to increase dramatically because they weren't competing with the vultures. This isn't only a problem in India. They've also, in some parts of Africa, they have what they call vulture restaurants. There's a new term I learnt recently. So again, animals are left out for the vultures to take. Why this is important, not only is because of the cultural significance and because almost loss of a species, is this was unexpected. This wasn't the kind of thing that people expected to happen, and we don't fully understand yet why. Now, it turns out that vultures are susceptible to diclofenac, but other bird species are not. Cats are also susceptible to diclofenac. So there's this funny trying to think of evolutionary pathways and why some organisms might be susceptible to some things and others aren't. Apparently there have also been issues in Spain with loss of vultures because, again, they leave animals out to be taken by the birds of prey. So what do we know for New Zealand? We don't really know very much yet. This is an expensive area, and people are working in this area. There are a range of different research groups around the country. What we do know is that some of these personal care products and pharmaceuticals, and only some of them, because we haven't looked for everything, are present in wastewater and environmental matrices at comparable concentrations to those measured internationally. So that's from work that I've seen other groups have done, as well as the work that's underway with my PhD students. There is growing interest from the regulatory agencies, so remember these are emerging contaminants. They're not on the usual monitoring lists, they're not being looked for. They only get looked for if you go out and do targeted surveys for them. So both Hawke's Bay Regional Council and Auckland Regional Council have commissioned some work on these, and encouragingly an AEE is an assessment of environmental effects. It's part of the Resource Management Act process. We're starting to see emerging contaminants and pharmaceuticals mentioned when people are applying to upgrade wastewater treatment plants. They say this needs to be considered, but of course, for many of these compounds, we don't yet have guidelines for what is an acceptable concentration in the environment. So it's kind of on the radar, but not really being dealt with. So how can we go about reducing our concentrations in the environment? Well, the US Food and Drug Administration now requires people to do some modelling for their active ingredients, and if the concentration is likely to be greater than 1 part per billion, then they need to do an environmental assessment for them. However, we've still got a large legacy of drugs that are out there that we have never done these assessments for. This one is a little bit going to take some doing. It's about reducing and restricting the use of pharmaceuticals. Definitely restricting the use of pharmaceuticals, I think, on animals is a good idea, particularly as we try and keep hold of or keep antibiotic resistance underway. Many people will be familiar with their GP not always giving them antibiotics, and some wait and see, or we'll give them to you later. Segregation of sources of pharmaceuticals. Now that ranges from whether some people, whether they're being treated with something, can only be treated in a hospital, and then that would go to a specific wastewater treatment plant, down to redesigning the toilet. Improving treatment of wastewater and drinking water is important. Many countries, the concern about these chemicals getting into drinking water has been the driver for research. Well, that's not the driver so far in New Zealand. This one's important, about using green chemistry, so improving drug design. So there are some groups where they bring in a toxicologist, so an expert in working out what effects might be in aquatic organisms, a design chemist, a theoretical chemist, and they actually work out whether they think something will be a problem at the design stage before it's even manufactured. And that may be something we need to do. Looking at improving formulations and drug delivery, can we find a way to give people less of a drug, but it's much more effective? Do they really need 300 milligrams? Can we give them 1? This one's important too around developing pharmaceutical return programs. So that's some countries, New Zealand isn't doing this currently, they encourage people to return their unused and unwanted pharmaceuticals. This one's also important, and one that I would really support, is regulating the use of antimicrobials in consumer products. Maybe we don't need antibacterial everything and antibacterial wipes and things in our homes. This here is a study done in the US where they used a whole lot of different antibacterial ingredients, and they did a study over about a year. What they did was they compared households that we use, they provided them with their cleaning products, they compared households that had antibacterial compounds in their cleaning products and households that didn't. And they actually found that there was no difference in disease between those houses. So what this shows is that for many of these antibacterial cleaning products, in a domestic setting or in a house, they are no more effective than using soap and water at preventing disease. So given some of the effects that we know these contaminants can have in the environment, or these ingredients can have in the environment, we really need to start questioning, do we need to be using some of these as widely as we are? We can also look at improving our wastewater treatment. However, if we improve our wastewater treatment processes, they tend to be quite expensive and they tend to be very energy intensive. And there's some areas in Australia where they have put in very advanced tertiary treatment plants to deal with pharmaceuticals and personal care products, partially because they are going to need to reuse that water, and particularly in drinking water. And they found the cost of them, particularly for smaller communities, is prohibitive to run them. So some of the options that we have are using ozone, using ultraviolet light, combining different oxidation processes to see whether we can actually come up with the magic bullet that will deal with them. But really this is an area where a lot of people are actually trying to work out what is the most practical option. And there is also the question of, does improved wastewater treatment mean the most high tech? And we're going to be involved in some work next year looking at different treatment options. This here is, along the bottom, are a range of different treatment options, so from sort of primary and secondary, like most of the wastewater treatment plants in New Zealand, using wetlands. Reverse osmosis, that's kind of you're on the way to treating it like you would for preparing drinking water, adding in ozone. And as you can see, what we've got here is the fraction of different chemicals remaining in the effluent, so what comes out the other end. What you can see is we've got quite variable levels of treatment between the different methods. Now this variability will depend on the compound and on the type of wastewater treatment plant. So we're still figuring out how do we re-engineer some very old technology which was designed to stop us from getting sick from pathogens to actually remove some of these other chemicals that we're now worried about. As I said, the US FDA requires an environmental assessment for human drugs. So does the European Medicines Agency. They now require that for human and veterinary pharmaceuticals. Anything you want to take to market, you now have to do an environmental risk assessment for. And this one is interesting. This is the first time these chemicals have been added to any kind of watch list. So the EU Water Framework Directive is around protecting drinking water. There was a big debate last year. They were actually going to introduce guidelines for ethanol estradiol, E2, another form of estrogen, and diclofenac. However, what they've decided is they've added them to the priority substance watch list. So they're now watching what kind of information comes out about these, and will be deciding whether these will now have to be looked for in drinking water in the future. Of course, we've been talking about legal compounds, and I should also point out that many of the illegal compounds, illegal drugs, are also found in the environment. The kind of regulatory approaches that we have been talking about probably won't deal with these. However, people are looking at concentrations of some of these illegal compounds in sewage and in the environment. You can use it around music festivals and things like that to look at the effectiveness of your policing. So did it get in? Did our latest strategy work? And it's also been used in some areas to target suburbs that might need a closer look, because you can tell where your sewage has come from and have some idea of where things might be worth looking. One of my students came back from a conference highly amused. A university had, and it's probably a good lesson, don't analyse anything that you own or you work out or anything like that, very good rule. One of the people had done some work looking in one of the frat houses on American University, and they could actually narrow the drug use down to a floor. The university didn't want them to publish that information because I don't think they thought that was a good recruitment tool with parents. Okay. So, in summary, yes, we are finding active ingredients from everyday products, including our personal care products, our shampoos and toothpaste, and pharmaceuticals in the environment. Some of these emerging organic contaminants are having adverse effects on aquatic organisms. Why this is important for you is because communities are increasingly going to be asked questions along the lines of, should certain high-risk chemicals continue to be used? Do you want to have antibacterial compounds to use in your kitchen, or do you not? And if you do, or are you willing to fund upgrades to wastewater treatment plants to perhaps prevent some of the effects that we're seeing in aquatic organisms? And with that, I'd like to finish. I apologise for the cough. Next time I'm going to take a better pharmaceutical.